There's no way.
Okay, thank you everyone for coming to Megha Makam's thesis defense. It's a pleasure for me to introduce her. I can honestly say that Megha has probably had the greatest diversity of research experiences of any graduate student I've ever known. Already when she was an undergraduate at Illinois, she worked on the chemical warfare between plants and insects, working on poisoned hemlock and moths that feed on poisoned hemlock. So you might even say that's a venture into the classics because of the poisoned hemlock. She also worked on a project investigating or using fMRI on individuals that were exercising. And I don't exactly know what the purpose of that was, but now she's coming full circle around to exercise again. And she then left Illinois and did a research internship in Ireland, studying the effects of crop herbicides on plants, comparing different herbicides, and then went to France and did a study of Drosophila and taste aversion in Drosophila, and then from there came to the Herzenberg lab. I'm really pleased that Lee Herzenberg is the chair of the thesis committee, because she introduced Megha to Stanford. And in the Herzenberg lab, she, of course, used flow cytometry and did a very interesting project on activation of neutrophils in the lungs of cystic fibrosis patients. And that ended in a first authored PNAS paper. So that's even before she started as a graduate student. So then she came to biology, and she was eager to expand her experience into animal work, leaving cells behind. So she initially did a project with a postdoc in the lab, Asya Rolls, an amazing project which showed that we could actually modify memories during sleep. We could intensify memories, or we could erase memories or decrease them. And I thought that was an amazing beginning for a terrific thesis. And she then told me that, well, she really didn't know if she wanted to continue to work with animals. So I said, well, in your wildest imagination, what would you like to do? And she brightened up and said, neurobiology and music. So we made an arrangement with the music department, Jonathan Berger, the chair, and she started to do projects over in the music department, and it was looking positive because they were getting a new faculty member who actually did neurobiology and music. Megha did several projects, traveled to conferences and presented her work, and then presented to her committee her thesis proposal, which was a beautiful piece of work, but we all thought it was overly ambitious. It was not a thesis proposal, it was a lifetime career proposal. So we asked her to go back and start small, take small steps first, and she did that. And then it became obvious that there was not going to be an easy task transferring from biology to music and doing biology and music. They didn't have the facilities, and there was not the funding available. So after a little bit of frustration, she came back to the mice. But the mice didn't cooperate. So then she got her final wish to work with humans, and that's the work she's going to tell you about today, following up work that has been going on in our lab for a long time in being able to manipulate the heat content of humans. Thank you.
Thank you, Craig, for that introduction. I forgot about a lot of that stuff. Good morning, everybody. Thank you for being here.
Good morning.
It's really nice to see so many familiar faces and surprising faces, and people I've known for a long time. So it's a pleasure and honor to be able to share my dissertation research that I've been working on the last few years. And this dissertation work was concerned with exploring the role of glabrous skin in human temperature regulation. Now, glabrous skin is hairless skin that we have on several parts of our body, for example, the palms of our hands, soles of our feet, and these are specialized regions for heat transfer. And so I went about exploring how we can use them and to basically exploit them to take advantage and treat and prevent hyperthermia in individuals, specifically wearing a highly insulating attire such as this firefighter. These individuals often wear heavy equipment, work, train, and perform in hot environments, oftentimes not able to take off the suit for long periods of time, and experience a thermoregulatory challenge where their core temperature starts to increase over the course of their work. So, for example, healthcare workers in West Africa treating patients of pandemics like Ebola, for example, are wearing gear that they must treat the patients in. However, over the course of their treatment, they experience an elevated core temperature and they need to take off the suit. However, every time they do this, they risk self-contaminating with the biological agents that they're trying to prevent themselves being exposed to in the first place. So this is one example of many occupations that could potentially benefit from being able to stay longer and work longer within the confines of those equipment. So the set of biological processes that help us maintain a healthy core body temperature is called thermoregulation. And we have a very narrow temperature range that mammalian cells function optimally within. And this function is a function of how much heat is in the body versus how much heat can be dissipated from the body. And we can describe this in an equation where on the left side we have metabolically generated heat as well as radiative heat that we absorb from the environment. On the right side of the equation we have radiation that we absorb from the environment, for example the sun's rays, and through convection and conduction, and evaporative cooling, for example why we sweat, perspire, and evaporation cools the skin surface. And all of these four variables are dependent on the surface temperature of the body. And this is because heat travels along a gradient from higher heat source to a lower heat source. And the temperature difference between the body's surface and the surrounding environment determines the rate at which this heat will dissipate from the body. So when there is an imbalance such that heat starts to get stored in the body, we experience an elevation of temperature. So after some intense exercise, and especially in a hot environment, core temperature starts to elevate. And soon after, we see a degradation of cognition, and we term anything above a core temperature of 39 degrees as hyperthermia, at least in this talk today. And further beyond that, we have pathological symptoms such as heat stroke or brain lesions. And if these conditions are sustained too much longer, they can be oftentimes fatal. So how does the body maintain this very narrow temperature range? Well, mammals have adapted to having a variable ability of dissipating heat from the body's surface. So we have a picture of an elephant here. And one elephant is in the water and the second elephant is on land. And we have an infrared image of the same picture of elephants. And what we see here is we can predict, by looking at the brightest spots of the picture, the thermal balance of the animal. So here we have a brightly lit trunk and ear, and on this animal we have a darker ear and kind of mottled and darker trunk, indicating that the animal on land has a lot more heat that's dissipating. Meanwhile, the animal in the water has a lot less heat to be dissipating to the environment. And in humans, we also have the specialized regions of skin that help us dissipate heat, that are optimized to transfer heat to the environment. And on this image, this infrared image of this runner, we have a very bright face, especially in the cheeks and on the palms. And these regions are called glabrous skin. These hairless regions of skin on the body that are optimized to dissipate heat to the environment. They together comprise a very small percentage of total body surface area, but they can have a very collective large effect on core body temperature. So blood is the distribution media of heat in the body, and these surfaces are specialized and have specialized vasculature that allow this heat to flow beneath the surface. The first structure is called arteriovenous anastomosis. And this is a structure that is a muscular shunt, basically allowing blood to flow from the artery directly to a vein, bypassing nutritive capillaries that would normally supply nutrition to other tissues. So during a thermoregulatory response, or when there's need to exchange heat, the valves change diameter, allowing different levels of blood to flow between the artery directly to the vein. And once it enters the venous side, it enters a structure called the retia venosa. And this is a highly complex network of vessels that accommodate a large volume of blood. And these are immediately after the arteriovenous anastomoses. And because they lie just beneath the surface of the glabrous skin, they allow very easy contact and heat to transfer across the surface of the skin. And you can see this during times when a large amount of heat is generated within the body. So during exercise, we see an elevation in blood flow underneath glabrous skin. So in this IR image, we have an individual before and then during exercise. And during exercise, you see a bright spot generated at the cheeks, and also in this individual at the cheeks and at the palm, compared to before, where it's much darker. And to take a closer look at the vessels that are responsible for this response, you can see the palmar side of this is a scanning electron micrograph of the palmar side of the thumb, of a human thumb, and we have this very dense network of vessels that comprise the retia venosa. Meanwhile, on the dorsal side, so it's the back of the hand, we have a very sparse network of vessels, and the ones that are there are actually just there to have nutritive functions, serving sweat glands and hair follicles. So the AVAs are, as I mentioned, a shunt that regulate blood flow between an artery and the vein. And they follow Poiseuille's law. And according to this law, it states that the flow rate is proportional to the fourth power of the radius, which essentially means very small changes in the radius of the vessel lead to very large changes in blood flow. So if this blood flow can change underneath the glabrous skin, it can change according to the needs of the organism. And to illustrate this point, a vasodilation, or an expansion of that radius of the vessel by about 19%, yields a doubling of the rate of blood flow. Meanwhile, a vasoconstriction, or a narrowing of that vessel by 16%, leads to a 50% reduction in the rate of blood flow. And so we ask ourselves, why can these small, specialized regions of glabrous skin have such a large effect on core body temperature? We have a video here of an individual who's exercised and then placed their hand in a very cold container of water, and the blood in the hand has cooled down, and that cool blood now travels up the vein of the arm. So if you can see the dark going up, and hot blood, warm blood, coming down the artery into the hand, you can see the hand rewarming at that region. So this, a couple decades ago, gave our lab an idea to take advantage of this system that we already have in place. So we start with a chamber, and we place a water convection pad inside, and it's constantly circulating cool water at 16 degrees Celsius, for example. And we place a source of a vacuum, and when there is a seal, there is a negative pressure inside that chamber. So we place the hand inside. This is the hand with the palm over here. And the warm blood enters the palm through the arteries. The blood enters the retia venosa, as you saw here. The heat transfers across this skin interface with the water perfusion pad. The water whisks away through convection the heat transferred from the hand, leaving that blood cooled. And that cooled blood then returns back up the vein to the heart. And in reality, this is what this device looks like. And this is the main workhorse in our lab right now. So we start with a chamber, and we create a vacuum by placing a cuff around the wrist, and the palm rests on the surface of this water perfusion pad inside. And this is the circulating water bath. We can set the temperature of whatever water temperature you'd like. And so the lab has shown that it can use this device and this principle of accessing the core, the body core, through the Glabrous skin portals. So we can either warm or cool the body by inserting or removing heat from those regions of skin. And we've been able to show that cooling through the Glabrous skin enhances work volume and improves strength training effects. We've also been able to show that cooling is able to improve aerobic endurance, as well as rewarm hypothermic patients after surgical anesthesia. And hypothermia is a common side effect of anesthesia and is a common problem actually in hospitals to rewarm patients after surgery. And these studies have all served as the foundation for the work that I'm about to share with you for my dissertation work. The first study was concerned with quantifying the rate of heat transferred from the glabrous skin compared to non-glabrous skin regions to understand how is glabrous skin different from non-glabrous skin. Second, I applied these principles to manipulate core body temperature of individuals while they're wearing highly insulative clothing. In this case, we used PPE, or personal protective equipment. And we used a variety of ambient temperatures and workloads to test the capacity of removing heat from the body core. And finally, we further took these ideas and applied it to testing whether we can manipulate or change people's level of thermal comfort inside a everyday office environment. I'll be sharing a few different experiments here, and a lot of them use similar methods of measurement, so I'm going to review them now. The first is esophageal temperature, which is measured by inserting a probe through the nostril, and the probe comes down the back of the esophagus and rests beside the heart. And so you can see this in this little diagram. It is as unpleasant as it sounds. And the heart rate was measured by a chest strap monitor and a wristwatch data logger. And I'll be presenting the data in terms of cardiac drift, which is the rate of change in heart rate over time. And cardiac drift represents the elevation, the constant elevation of heart rate in response to a fixed load exercise. And what we are doing in all of our studies is setting a very consistent rate or intensity of exercise for the individuals to complete. But we see a constant increase in heart rate, and this is a normal human response. And it parallels the rise in core temperature. In a couple of these studies, I measured heat flux. And heat flux is essentially the amount of heat transferred between two surfaces, and often using the skin of the glabrous skin of the palm or non-glabrous skin region. And the second surface would be a water perfusion pad, for example. And the data figures I'll be showing you have error bars, which are representing standard deviation, and the stars indicate the level of statistical significance. So 3 stars indicating the most significant. So the first study that I completed was quantifying the rate of heat flow from non-glabrous skin versus glabrous skin. Core temperature elevates quite rapidly through that exertion. And we wanted to find a way to treat these individuals faster than a conventional method. And so the gold standard for treating hyperthermia in the field for athletes is full body immersion in a cold container of water, or even in a river. However, that's usually not handy, and so the next best method is wrapping a cold ice towel around the neck. And the thing about that method is that it hasn't really been shown to be very effective, despite its being used for many, many years. So we went about asking whether glabrous skin cooling is more effective than a conventional method of exercise-induced hypothermia. We simulated the conditions by having individuals wear PPE suits and walking on a treadmill at a fixed workload until they reached a core temperature of 39 degrees. And the ambient temperature of this environment was 36 degrees. After completing that exercise and reaching 39 degrees, they would rest in the same environment for 30 minutes. On 5 separate days, so we had 5 different conditions. And the first condition, in the control condition, the subject's hands were wrapped in plastic bags to prevent evaporative heat loss, and no cooling was applied. In the second condition, a cold towel was wrapped around their shoulders and neck, and also placed underneath the PPE suit. And this was replaced a total of 3 times, so the fresh towel was always cold during the course of the entire 30 minutes. In the next condition, one hand was remained untreated, and the second hand was treated by placing it in a cooling device and engaging the vacuum. And so the idea of the vacuum is to promote the expansion of those vessels that I mentioned, the retia venosa, to fill with more blood, allowing more heat to transfer from that region. In the last 2 conditions, we cooled both palms, but one of them did not engage the vacuum. So we wanted to see how effective the vacuum was as well. And what we show is that when no cooling is applied, over the course of 30 minutes, core temperature continues to rise, even if they're sitting still. Any cooling method that's applied has some effect by reducing the rate of core temperature rise. However, cooling both hands is the most effective. And we see this by comparing the ending temperature after the 30-minute rest period. So the 2-hand cooling method yielded the ending temperature at the lowest value. And we compared the mean rate of core temperature rise with a control compared to all the other conditions. And so you can see that during the control condition and no cooling was applied, core temperature rose in a positive rate. And in any cooling condition, core temperature changed at a negative rate and reduced. And in the last 2 conditions, we actually didn't find a significance between 2-hand cooling with and without vacuum. And that indicates that during a hyperthermia response, those vessels are maximally dilated, and adding a vacuum must not actually add too much more of an effect on expanding those vessels. So we were able to answer the question, is glabrous cooling more effective than a conventional method to treat exercise-induced hyperthermia? And we show that glabrous cooling is a faster method than cooling with a towel. The last part of this section of studies asks the question whether glabrous cooling can delay hyperthermia in individuals sitting in a hot environment while wearing PPE. And we are revisiting the same protocol that I mentioned at the very beginning of this talk. And as a reminder, individuals wearing full PPE gear rested in a room at 45 degrees Celsius until their core temperature reached 39 degrees. And we simultaneously measured esophageal temperature and heart rate. And what we show over the course of the 60-minute trial, on average, that's how long it took for individuals' core temperature to rise until 39 degrees. Some of them took longer, some of them were faster. However, if we take a look at the ending temperature of around 39 degrees, and they both started around the same, we see a significantly lower ending temperature when we cool the skin, the glabrous skin of the palms. There's barely over a half a degree increase in core temperature. And we see that if we compare the rate of temperature rise, here we see a comparison of cooling versus not cooling, and temperature changed at a much lower rate when cooled. Similarly, polymer cooling had an effect on reducing cardiac drift as well. So we were able to answer this question: can glabrous cooling delay hyperthermia in subjects wearing PPE and resting in a hot environment? So to summarize this section of studies, we showed that we can manipulate the core temperature of individuals wearing PPE and working at various workloads in a variety of environments. We showed that we can attenuate the rate of temperature rise during exercise, aerobic exercise. Also, we can have similar effects using a wearable cooling device underneath the suit. And we can use glabrous skin cooling as a better method or more effective method than a traditional method to cool hyperthermic individuals after they've completed exercise. And we've also been able to show that we can delay the onset of hyperthermia by glabrous skin cooling while they sit in a hot environment wearing heavy gear. And these studies have major implications for individuals wearing protective equipment such as Ebola workers, and completing a very low workload, exercise workload, or nothing at all, resting, actually would give us a good idea of how much time they might actually have working in that suit, and how much time we can extend their working time. So the last section of experiments deals with delivering or providing thermal comfort to individuals resting in an environment that's more commonly found in office buildings, for example. So we asked whether we can use glabrous cooling to help affect their thermal comfort. And this study was motivated by the Department of Energy's ARPA-E call for the development of a personalized or individualized cooling system in order to deliver or provide thermal comfort to individuals in an office environment. Now, this was motivated because they're trying to overall reduce the amount of energy consumed. An association of heating and cooling engineers have established many years ago that the range of ambient temperatures that are most comfortable to people in an office environment is between 20 to 24 degrees Celsius. However, this call for the development of more technology wanted to extend this range out to 27 degrees before a cooling system would be engaged. By extending the range of ambient temperatures people are more comfortable in, the HVAC system, and especially in this case the air conditioning system, can be used at only higher temperatures, which would overall allow less energy to be used. So we created a scenario in the lab where individuals would rest in a chamber set at an ambient temperature of 27 degrees Celsius. We had two conditions. They would wear either clothing, summer clothing of their choice, which was usually a shorts and a t-shirt, and the second condition was wearing a hoodie to increase the insulation layer of that clothing. Their bare feet rested on water circulation pads, and if you recall, glabrous skin is also on the bottom of the feet. And so we have room temperature water circulating through those pads. We also measured heat flux. So as you recall, these sensors measure the transfer of heat between two surfaces, And so we sandwiched a sensor in between the pad and the skin of the foot, allowing us to measure how much heat is transferred between the two surfaces. And over the course of the 50-minute protocol, or the trial, each subject would complete a survey, and this is a standardized survey of thermal comfort established by this Association of Engineers. And they rated a 0 if they felt neutral, 3 would be the most warm, which is hot, 2 would be warm, 1 is slightly warm, etc. And what we showed is that they're not actually that uncomfortable in that environment. So when they wore summer attire, they experienced a near neutral rating of thermal comfort, and wearing the hoodies elevated it to around 1.5. It's just above slightly warm. And with cooling in each condition, there was no difference found in how their thermal comfort ratings. And secondly, we measured heat flux, and this also kind of explains why they might not be uncomfortable. So heat flux is the measure of heat leaving the body, in this case, but we don't see an increase when we apply a thermal gradient or a cooling pad, meaning there's not a lot of extra heat to leave the body. So whether they're wearing summer attire or hoodies, we don't see any difference in the amount of heat that they're dissipating from the glabrous skin of their feet. So we tried to build on this experiment to say, well, we need to make these individuals more uncomfortable, first of all. So what we did was added 2 more conditions where we had warmed water circulating through the pads and also cooled water circulating through the pads at 17 degrees. And we added 1 more condition. In addition to the hoodies condition of insulation, the heaviest condition was wearing our favorite PPE equipment. And they continued to rate their thermal comfort on the scale during the course of the trial. And so this time we found that they eventually got uncomfortable when we put on the heaviest layer of insulation, the PPE equipment, and also had to circulate warm water at their feet. But until that point, they did not really feel all that uncomfortable. And so this is a very strong illustration of our misunderstanding of how our thermoneutral zone resides. In other words, thermoneutral zone is the range of temperatures at which our body is most comfortable and engages the least thermoregulatory effectors. So we don't sweat, we don't shiver, we're feeling good. And the Department of Energy and the general public accepts that value to be between 20 and 24 degrees Celsius. But through this study, we found that's likely looking more like this, and it could extend further out. So the range of temperatures in which we're more comfortable is quite larger than we think. And by showing that applying cooling to individuals, once they are uncomfortable, we show that we have potential to actually influence people during different seasons. So during the winter time, when the ambient temperatures drop, we can actually provide comfort at lower ambient temperatures. And during the summertime, when ambient temperatures rise, we can provide thermal comfort at warmer ambient temperatures. And this would have major implications. By using less energy to treat the air, we would overall use less energy as a whole. So buildings actually make up about 40% of the total energy expenditure in the United States. And of that, about a third of that is devoted to HVAC systems, so delivering thermal comfort to individuals and buildings. And so just small changes in the practices of engaging the HVAC systems in these buildings could actually have major effects. And so to summarize the last section of studies, I showed that individuals need to be sufficiently uncomfortable before we can manipulate their thermal comfort. but that it is possible once they're at that point. And that thermal comfort curve, as I showed you, that zone of thermoneutrality is likely much wider than we expect. And by modifying our behaviors and engaging in less engaging the HVAC systems at wider ends of the energy spectrum of the thermoneutral zone, we can actually have major impacts on energy expenditure. So finally, in this section, I was able to show that, yes, we can exchange heat through glabrous skin in order to deliver thermal comfort, but only when people are sufficiently uncomfortable. So to summarize the work I've just shared with you, I started by quantifying the rate of heat transfer across the glabrous skin and compared it to non-glabrous skin to show the difference between the function of the two skin types. Second, I used these information to change the core body temperature of individuals wearing personal protective equipment and working in different ambient temperatures and at different workloads. And finally, I was able to explore whether glabrous skin manipulation can also affect the rating of thermal comfort in normal office environments. So to summarize overall, I have been really grateful to be able to work on a topic that actually has immediate implications for individuals that are also similarly trying to make a big difference in other people's lives. So I feel grateful for that opportunity, and I've had a really nice time getting to know how the body works and all the applications that this work can actually have. So at this time, I would like to acknowledge a lot of people that made this work possible, but first I will take some questions while they might be fresh in your minds. So thank you, and I'm happy to take some questions. Hannah.
Great job!
Thank you.
Did you notice any sex differences?
We did not.
Repeat the questions.
Oh yes. So Hannah asked whether we noticed any sex differences, and we did not. However, we did not have the numbers to do a full statistical measure, but that would be something interesting to look at. Teddy.
I had a question about perception of temperature response. So like an athlete running around outside in the heat, you know, they're hotter, but they wouldn't be concerned about it. Whereas somebody in the office, the temperature goes up 2 degrees, they're whining about it, right?
Yeah.
Did you kind of factor in some of the psychosomatic effects? Or?
So that's a really good point. So Teddy asked about the perception of the ambient temperature depending on the context. So comparing, for example, an athlete doing exercise and kind of ignoring that input, whereas an office environment, someone might be easily uncomfortable. We did not particularly control for that exact question, but there is definitely a documented effect of perception of the thermal environment. So it's well known and documented in studies that show that athletes are able to push through thermal discomfort, and that actually is why we see some oftentimes fatalities in athletes that can turn off that danger signal inside when they get too hot. So in terms of the thermal comfort study we did not factor in that particular question, but it is definitely a factor. Kids.
Do you feel like if they put the towel on the hands instead of the shoulders, that would be the remedy that you're looking for in the absence of the machine?
Sure, that's actually a very good point. So Kit asked whether placing the towel on the hands instead of the shoulders would actually be a better way to go in terms of treating hypothermia. And actually, a similar study has been done by placing, chemical cold packs that are used, for injuries instead of placing on the traditional areas, which are like the axilla, the neck, the groin, they've been placed on the glabrous skin regions and shown that that has a greater effect than cooling through those more traditional regions. So to answer your question, yes, cooling using whatever you have on hand, pun intended, I don't know. So you should be able to see a little bit better effect of cooling, yes. Bill.
Are there implications for dehydration using this method compared to blowing sweat off with a fan and losing a lot of water?
Yeah, the implication is that less water loss would be resulting from cooling through the palms or the feet. Yes.
So intuitively, I would think that the surface area of your hands is relatively small. The heat flow is kind of the product of the heat flux of the area.
Yeah.
What's the ratio of the difference? Like you had this graph when you were looking at it. What's the ratio of that difference versus, say, I was thinking these wearable ice backpack, whether you could just have big ice thing that just touches your entire back.
Okay, so if I understood the question, you're asking the difference between the surface areas that are treated by the Glabrous treatment versus the entire back, for example. So the key to this is actually the difference in the vasculature underlying the glabrous skin. So the back doesn't have these, the skin of the back doesn't have these specialized regions or specialized circulatory structures that bring more blood flow for the specific purpose of heat exchange. So though it is a greater surface area, you would most likely be just cooling the local tissue, but not actually accessing the core blood. So the surface area is a secondary kind of variable to the blood flow that you're actually accessing.
At some point, you would rather be...
So instead of, yeah, so of course, like that's why full body immersion works. At some point, there is a balance. So if you cool the entire, I don't know exactly the exact ratio of how much of the total body surface area you could, like non-glabrous surface area compared to just a small percent of the glabrous. That ratio I do not know. Thanks.
How do you interact temperature and perspiration? One would think that your hands don't perspire as much, or do they? I mean, what's the story?
The story on perspiration during hyperthermia or in general?
Hypothermia.
Okay. So the hands aren't specialized to perspire in response to heat. The glabrous skin of the hands, that's more of a body surface response. So it certainly contributes to heat exchange. So as cold breeze blows over a sweaty palm, it does transfer a good amount of heat. In addition to, and it just enhances the heat transfer that's already occurring based on the increased blood flow we also find at the palms. But I don't know if, yeah.
But mainly what I'm asking is there less heat loss due to perspiration on the back or the hands?
Okay. So if the back were sweaty and the palms are sweaty, which one? If we were measuring the same amount of surface area, it would be the hands. If they were both the same amount of perspiration and the same amount of surface area, the hands would lose more heat. Yeah.
I'm curious, if other animals, like in birds that have a brood patch, can they automatically regulate the transfer of heat from that when they're incubating eggs, for example? They warm them up because they have this glabrous patch there. Do
Yes.
they have a different set of structures than we do?
I'm not aware of particular specialization that birds might have, but if they do have these circulatory structures underlying that skin region, if it's hairless, and it would transfer more heat. Yeah, it all depends on the circulation at that region.
So in the last set of experiments with thermal comfort,
Yeah.
From the pictures, it looked like these were mainly college-age students who were
Yes.
being involved.
Yes.
So since since the average BMI of the general office worker in the United States is significantly different from that cohort of healthy Stanford undergraduates, do you think that that might contribute to a dichotomy between your findings and the generalizability of those findings of thermal comfort?
There might be. Yeah, I would expect there would be an effect of increasing the average BMI of the subjects.
Do you think it would be more difficult to lose heat through non-traditional sources? So I guess what I'm asking is, do you think that the specific cooling of non-glabrous skin, the non-hairy skin, would be more effective than, say, surface cooling in someone who is obese rather than someone who is not obese?
It would be, I think, especially effective due to those regions of skin they don't have the adipose layer that would normally be insulating. So, in fact, I would expect that the effect of glabrous cooling would be greater in those individuals if all other variables were constant, because of the ability to, compared to like a cooling with a fan or other non-glabrous cooling method. Yeah. All right. I think at this time I would like to move on to the acknowledgements portion. So I have so many people I'd like to thank, and starting with people in our lab, I can't express how much I appreciate all the times you have done pilot experiments upon pilot experiments and sweating literal buckets for science, and sticking that probe up and down all the time, and watching out for each other as well, making sure everyone's healthy. So that has been great. And the committee members, I appreciate you for all the years of your help and advice and support through all of these different projects. Thank you, Lee, for chairing this day. It feels really nice to come full circle in terms of my time at Stanford and have you here with us today. Thank you, Dennis, for all this help developing the systems and showing me the way through all of the questions, the dumb questions I've had over the years, trying to get up to speed really quickly. And Craig, thank you for your support. As you mentioned, all the ups and downs I've had and scientific adventures, you've always been supportive and understanding and patient, so I really appreciate that. Thank you, Samantha, for all your help. Dan's not here, but student services staff has been amazing, keeping us in line and making sure we're always on top of our stuff, and it's very much appreciated. And a really big shout out to all the volunteers who put themselves through all sorts of things. Some of you are here today to make this work happen. Without volunteer subjects, this work would not exist. And so I have 2 more slides. The first is, yes, this is Portuguese. So I had to share this poem with you today because about a year ago I took a trip to my happy place, Brazil, Rio de Janeiro, and this photograph was taken on New Year's Eve on Copacabana Beach, and it kind of represented a moment where I was transitioning in my mind about many things in life, and I needed to make a decision about my journey in science. And right after I came back and I decided to take on this new project, my friend sent me this poem, and I met her there, and she's also studying Portuguese literature, so I didn't read this on my own. But to loosely translate, it says, the title is Science. It says, a hypothesis. Happiness is a catalyst for a scientific experiment, and sadness an inhibitor. Sadness makes you shrink, so how can a sad person discover anything? Only happy people take risks, and sadness is anti-scientific. So I had to share this because points in one's career, not just in science, but you might have these moments where you need to evaluate things, and sometimes you just have to read some poetry. So with that, I will say thank you.
May I ask you one more question?
Yeah.
How does a drummer's hands deal with heat exchange, calluses and things like that?
Actually, I would like to study that question. Calluses probably increase the insulation barrier in those parts of the hand. Usually drummers don't have calluses over the entire hand surface, so luckily they have some heat exchange available, but calluses would create a little bit more of a barrier between that particular region of skin. So with that, thank you, and I really appreciate you all for being here today. I guess this is the end of my talk and the start of a private exam session.
And there will be a lunch outside of Gilbert at 12:30 for anybody who wants to come.
You do.
Good job.